I'm Justin.
I'm Christiana, and we did a project at the Energy Lab that deals with fast twitch muscles and basically just doing a bunch of biology.
So basically what our experiment is we take DNA by swabbing people's mouths, and we replicate that DNA in here, and then we show it up on a gel, and then we look at the gel on here, and we can see whether a person has a particular gene or not.
We decided to do this project because we're all really interested in biology and genetics. We've taken an AP Bio class together and we're looking to forward our studies.
And so we chose this particular project about the ACTN3 gene, which is a gene that codes for fast twitch or slow twitch muscles. And so there's two copies of every gene you have, and if you have the non-mutated copy or two non-mutated copies, you make a lot of fast twitch muscles, which fire really rapidly, but they tire really rapidly. So that means you can sprint, but you can't sprint for a very long time. And then if you have two mutated copies, that means you don't make very many fast twitch muscles, so you can run for a long time, but you're not necessarily going to be a very good sprinter. And so we wanted to see if the type of gene that somebody had in our school affected what kind of sport they play. Because at the Olympic level, they've done studies and they show that if you have two of the mutated alleles, that you tend to be a really good distance runner. But if you have two of the normal kind, then you'll be like Usain Bolt. And so we wanted to see if maybe the good sprinters at our school had those two fast twitch muscles, non-mutated copies, and maybe the good endurance runners had the two mutated copies.
So basically we tested that not only with runners, but with swimmers, basketball players, and also people who do no sports at all, just to see if there's any correlation between doing sports and that gene, or if it's just completely random at the high school level.
So after getting our DNA, basically the thing that we had to do is we had to make a bunch of copies of it, because you can't really do much with DNA if you don't have a bunch of it, because it's really, really small. And so what we would use is this, which is a thermocycler, and it was actually built by the company that an HPA student started. And the really cool thing about this is very, very few high schools have a machine such as this. They cost thousands of dollars and they're really expensive, but this one is a cheaper version of it. It's still a really good version of it, it's just you have to make it yourself. And so we're really lucky to be able to work with it. And so we load our DNA into here, and we hook it up into the computer, and if it will turn on. Basically, this is the computer program that works with it, and there's a bunch of different experiments that's stored on it, and there's cycles, and the cycles will denature the DNA, basically pull it apart, and then make new copies of it. And every time you run one cycle, you get double the amount of DNA. And so if you run 30 cycles, you end up getting 1 billion copies of this one little segment of DNA. And so after running this, we have 1 billion little copies of DNA, and then we'll load it into a gel electrophoresis chamber, and we will.
So basically what we do with that is we make these little agarose shells that are tiny little squares, and they have 8 pockets in them, wells we call them, and we use the pipettes that are fancy. They're really specific. They go down to 0.1 microliters we have. So basically what we can do with that is we take the DNA that we just copied in here, and we put it into the wells that we have, and we put it into a giant electrophoresis chamber like that. And a voltage runs across it and pulls the negatively charged DNA towards one side. So basically what this ends up is we get bands of DNA across it, and we look at it under here in usually a darker room, and with these orange sheets and goggles, it cuts out all the blue light so we can just see the DNA that's glowing green from it. Yeah, from a stain that we used while we were cycling it. And we can see whether that subject has a gene or not based off of what we see.
Yeah.
All right.
Yeah,
So,
is that about it?
yeah.